Hey there again, fellow scientists. Today we're diving into 1.6 to 1.7, so let's get to it. And I think we should start with nucleic acids, the information carriers, DNA and RNA. Their monomer is the nucleotide, which has 3 parts, a 5-carbon sugar, a phosphate group, and a nitrogenous base. Nucleotides link via dehydration synthesis to form a strand with directionality. And this is definitely important and a little bit complicated, so pay attention. The double helix of a DNA molecule contains 2 strands, one running from 5' to 3' and another running from 3' to 5'. This, my scientists, is called anti-parallel. And you also need to know that DNA has directionality, meaning that it is only synthesized from 5' to 3', not the other way around. Directionality results in a lagging strand and a leading strand. Now that we've got that down, we need to talk about the base pairs. The 4 base pairs of DNA are A, T, G, and C. Well, what is one to do with all these letters? Well, you need to remember how they match up. A pairs with T, and G pairs with C. In contrast, RNA is typically a single-stranded molecule using the sugar ribose and has uracil instead of the letter T, thymine. This simpler structure of RNA allows it to play different roles in expressing genetic information, ultimately making up proteins. So nucleic acids carry the blueprints, but what actually makes these blueprints into life? Proteins. Proteins are the workhorses. Their monomers are amino acids linked by peptide bonds, remember, and the most important part of amino acid is its R group. The R group distinguishes one amino acid over the other. Now, let's think bigger. The entire function of a protein is determined by its structure, which has 4 levels. Level 1 is the primary structure, the specific linear sequence of amino acids. This sequence dictates everything else. Now onto the secondary structure. This is where things get a little bit more complicated. This involves taking the amino acid chain from the primary structure and folding it into patterns, such as the alpha helix, a coiled shape held together by hydrogen bonds, and the beta-pleated sheet, a folded sheet-like structure also stabilized by hydrogen bonds. Next, we have the tertiary structure. This is the overall 3D shape of a single polypeptide, formed when secondary structures fold further due to interactions between R groups. Now, the tertiary structure is where some proteins are made, but other proteins go on to the quaternary structure. The quaternary structure involves several interactions between tertiary structures, basically forming one big protein. And have you ever heard of the word mutation? Well, of course you have, but do you even know what it means? Well, a mutation is a change in an amino acid in the primary structure, which can completely change the function of a protein. And that is your rapid fire review of nucleic acids and proteins. And don't forget the golden rule, structure determines function. Now, before you smash that in your head, smash the like button and subscribe for more bio breakdowns. I'll see you in the next one.